First, the process begins at specific locations on the DNA molecule called origins of replication. Here, the enzyme helicase plays a crucial role by unwinding the double helix structure of the DNA. Helicase works by breaking the hydrogen bonds between the complementary base pairs, effectively unzipping the DNA strands and creating what is known as the replication fork. Once the DNA strands are separated, the next enzyme, primase, comes into play. Primase synthesizes short RNA primers on each end of the single-stranded DNA templates. These primers are essential because DNA polymerases, the enzymes responsible for synthesizing the new DNA strands, need a starting point to begin their work. After the primers are laid down, DNA polymerase binds to the primer and starts adding nucleotides that are complementary to the template strand. It's important to note that DNA polymerase can only add nucleotides in one direction, from the 5' to the 3' end. Two strands are being synthesized during DNA replication, the leading strand and the lagging strand. The leading strand is synthesized continuously in the same direction as the replication fork because it can follow the unzipping action of helicase without interruption. In contrast, the lagging strand is synthesized discontinuously because it runs in the opposite direction. This strand is built in short segments called Okazaki fragments, each initiated by its own RNA primer. Once DNA polymerase has completed the new strands, another enzyme called exonuclease removes the RNA primers. The gaps left by the removal of these primers are then filled in by DNA polymerase. Finally, DNA ligase is responsible for sealing the nicks between the Okazaki fragments on the lagging strand, resulting in a continuous DNA molecule. End product of DNA replication is two identical DNA molecules, each composed of one original strand and one newly synthesized strand. Thank you for listening to our presentation about DNA replication.